Okay, so welcome to this next video in which we are discussing circadian rhythms. Okay, so in the previous videos what we have seen is that humans have these two nuclei in the hypothalamus called the suprachiasmatic nuclei, and that these two suprachiasmatic nuclei are effectively internal clocks within the brain. Okay, they have cyclical patterns of activity, and their activity is higher during the day and then plummets during the night. Okay, so basically they are the internal clock, and they are responsible for many of these other physiological parameters which also show a circadian rhythm of oscillation on a 24-hour period. Okay, but the question then arises, does this clock completely match 24 hours? Okay, so is it actually going to oscillate perfectly within 24 hours? Well, the answer is of course not. It's going to be close, but it's going to be slightly off, and that would mean that if it remained like that for a long period of time, eventually it will become completely out of phase with the real time, okay? So what we need to do is have a way of making sure that our internal clock is always in synchrony with the real world clock, okay? So in synchrony with the Zeitgeber, and the Zeitgeber that we're specifically interested in is the light-dark cycle, which occurs every day. Okay, so what we need to do is make sure that when the world is light, basically, when it's light, we have the higher activity in our suprachiasmatic nuclei, and when it's dark, that's when our suprachiasmatic nuclei are at their low points in that activity level. Okay, so that means that we need to have some way for visual information to get to the suprachiasmatic nuclei to affect the position of this cyclic activity level, basically. Okay, so what we now want to discuss is the pathway by which photo information is conveyed to the suprachiasmatic nuclei. And for this, we're going to start by looking at the anatomy of the eye, because within the eye there are special ganglion cells which are photosensitive. Now, ganglion cells are not usually photosensitive. Okay, usually they just convey the information from the photoreceptors, which are the rod cells and the cone cells, but there are special ganglion cells which are photosensitive and which convey their information to the suprachiasmatic nuclei. Okay, so we want to have a look at the anatomy of the eye and the anatomy of the retina, okay, just to revise that so that we're all clear with it, and then we'll look at these pathways from the retina to the suprachiasmatic nuclei, whereby visual information about the current time of the world can be conveyed to the suprachiasmatic nuclei so that they can make sure that their clock is in tune with the external world's clock. Okay, right. So let's start with the gross anatomy of the eye then. So we'll draw a picture that is here. Okay, so we'll start off by showing the choroid, and then we'll put the sclera and the retina around that. Okay, so basically at the front we have the iris, okay, with the pupil in the middle. So this can be the iris muscles here. And then the iris is continuous with the choroid behind, but the choroid also has this ciliaris muscle here, or the ciliary body, which is going to have the suspensory ligaments dangling from it. Okay, and that's a little big. Let's take it back down to there, and then we'll make this into the lens. Okay, right. So we'll have a similar structure on this side. Okay, and then the choroid will extend backwards like so. Okay, so let's work on these things here so that I can correct for my mistake here. So let's put the lens here, okay, sitting behind the pupil. So I'm going to have to colour this in to correct it all nicely. Okay, so we'll colour in the lens in blue here. So all of this that I'm now colouring in blue, this represents the lens. Okay, and then we have these suspensory ligaments which hold the lens in place, and those connect the lens to the ciliary bodies. Okay, so these little extensions of the choroid inwards here, these are a muscle basically, but they also have secretory function. Okay, so with regards to them as a muscle, they're often called the ciliaris muscle. Okay, but with regards to their secretory activity, they're often called the ciliary body. Okay, so whichever one you want to use, either is fine. Okay, ciliaris muscle you would generally use if you're talking about it as a muscle. If you're referring to its action at secreting fluid into the aqueous humor, then you'd call it the ciliary body. Okay, then what happens is the choroid extends back round like this. Okay, and similarly on this side, but then it doesn't completely go round in a perfect sphere. Okay, it falls short right at the end. Then what we need to show in here is the retina. So next here comes the retina, which will line the internal aspect of the eye. Okay, and we'll have a similar structure on this side. And then the retina will be continuous with the nervous tissue of the optic nerve. So this is where the optic nerve is going to begin here. Okay, so this now is the optic nerve here. And there is this channel moving through the optic nerve here, which I'll be able to show better if I now colour this in. So, let's colour in the retina in yellow here. Okay, so all of this now being shown in yellow, this represents the retina, which is continuous with the neural tissue within the optic nerve shown here. Okay, like so. So this is the optic nerve down here, and then the retina. Okay, and that lines the internal aspect of the eye. Okay, so I'll just label both of those structures up then now. So this we've said is the retina, and this tissue down here is the optic nerve. And we're drawing the right-hand eye here, so the optic nerve is going medially towards the nose, and therefore this is the medial direction. The nose will be sitting here. Okay, so this is the optic nerve. Okay, right. Then, this portion that's outside of the retina, this then is called the choroid. Okay, so I'll colour the choroid in red here. Okay, so this is the choroid, and it's continuous basically with the ciliaris muscle, or the ciliary body at the front, and also with the iris muscle. Okay, so all of this is continuous here, and the same on this side over here. Okay, right, so that goes all the way back here, like so. So let's label up a few more things. We've got the lens at the front here. Okay, we've also got the suspensory ligaments which are suspending the lens. Okay, and those will be an annulus basically, because of course we're only seeing a two-dimensional cross-section here. In reality, this stuff is all three-dimensional, and the lens will be a disc-like structure, and then the suspensory ligaments will be an annulus that surrounds the entire lens. Okay, so these are the suspensory ligaments. Okay, right. Then right at the front, this muscle here is called the iris, and this is the muscle, the annulus of muscle, which surrounds the pupil in the middle. Okay, right. Now, that's not complete yet. There's another layer that goes over all of this, which is the sclera. So let's now put the sclera in, which is the white of the eye that you see when you look at an eye. Okay, so all of this is sclera around the outside. It also coats the optic nerve here. Okay, and then it will be the same on the other side. And then at the front, what happens is the sclera moves away from the iris and moves over it like this, okay? And this little cap that you have that doesn't sit right on top of the iris, but instead has a pouch of fluid between it and the iris, this is known as the cornea. So at the front, the sclera is continuous with the cornea, basically. Okay, so this is the cornea, and then around everywhere else you have the sclera. Right. The other thing we need to discuss is these chambers. So this chamber at the front here, between the iris muscle here and the cornea, that is called the anterior chamber. Okay, so this is the anterior chamber. Okay, now it is continuous via the pupil with another chamber, which is the chamber between the iris muscle here and then the lens underneath this chamber here. And that chamber between the lens and the pupil in front, and then the iris and the pupil in front and the lens behind is called the posterior chamber. Okay, so this is often something that people find confusing. They expect the posterior chamber to mean this chamber here, but in actual fact, this is the anterior chamber between the cornea and the iris, and then continuous with it via the pupil, you have the second chamber here, which is between the iris and the lens, which is the posterior chamber. So yes, they are the same thing. Fluid in the posterior chamber is in contact with the fluid in the anterior chamber, but they're called separate names. And what is the fluid that's within the posterior and anterior chambers? What's known as aqueous humor. Okay, and it's produced by the ciliaris muscle, or the ciliary body, and secreted into the posterior chamber, and then gets into the anterior chamber via going through the pupil. Now, this chamber here then, which is behind the lens and in contact with the retina, this is known as the vitreous chamber. Okay, and it's full of a jelly-like gloop called the vitreous humor. Now this isn't really a fluid, it's much more like a jelly basically. Okay, if you've ever done dissection of the eye, you'll know what I mean. When it comes out, it's a big lump of jelly basically. Okay, right. Now, the one last thing I want to mention is that in this canal that we have moving through the middle of the optic nerve, you are going to have arteries and veins coming up that. Okay, so you have an artery coming up here, and you also have a vein going down. Okay, now these are the arteries and the veins. Well, this is the artery and the vein which will supply the retina basically. So the artery will bring the blood in and the vein will take it away, and the names of them are the central artery / vein. So you've got the central artery and then also the central vein of the retina. Okay, so running in this canal, you have the central artery of the retina, and then you also have the central vein of the retina. Okay, right. So that's the macroscopic anatomy of an eye then now. Let's now look at the histology of the retina in more detail, because I want to remind you of the different cell types that are within the retina and the usual involvement of the ganglion cells in the conduction of the visual information to the brain. Okay, so let's now have a look at the anatomy of the retina. So what we're going to do basically is we're going to take a little cross section down the retina and look starting from the bottom and working our way up and seeing what the different cell types are basically. Okay, right. So we'll start right down here at the bottom then. So at the bottom, the bottommost layer of cells that is in contact therefore with the choroid underneath is known as the retinal pigment epithelium. Okay, now these cells are an epithelium and they're involved in recycling the retinal pigment, so they are very important in phototransduction. Okay, they're involved in taking the pigment that has been involved in the reaction with light and returning it back to the initial state so that it is ready to react with another particle of light again. Okay, so this is the retinal pigment epithelium, and this is the most peripheral portion of the retina, so it's the bottom portion. Okay, and for short, the retinal pigment epithelium is often abbreviated to the RPE for short, so R for retinal, P for pigment, and E for epithelium. Okay, then sitting on top of the retinal pigment epithelium, what you then have are the photoreceptors. Okay, so you have rod cells and then cone cells. So we'll start off with a rod here. Okay, now the reason that rod cells and cone cells are called rods and cones is because the actual apparatus that they have for detecting light is either in the shape of a rod in the case of rod cells or a cone in the shape of cone cells. Okay, so here are the actual portions that are responsible for phototransduction. Here is a rod. Okay, now it's not the complete cell yet, it's just part of the cell, but this is the rod of a rod cell. Okay, and here is the cone of a cone cell. Okay, in purple, and these will be in contact then with the retinal pigment epithelium that's at the base here. Okay, then here is a little axon which links the rod of the rod cell to the cell body of the rod cell, and then the rod cell will also have another axon leaving it, which will go off and synapse onto the next neuron, and it will be the same sort of architecture for the cone cell here. Okay, right, so these portions down here are the actual photoreceptive portions. These are the portions which are actually going to respond to light. So they will often be called the photoreceptors, or the rods and the cones. Okay, now, separating the rods and the cones from their cell bodies, there is a membrane through which these axons that connect the rod and the cone to their cell bodies will have to penetrate. Okay, and this membrane is called the external limiting membrane. Now, there's also going to be an internal limiting membrane, and the reason this is the external one is that this faces the most externally. The internal one is going to be right on the internal aspect of the retina, facing the vitreous chamber, whereas this is quite far out. It's just after the photoreceptive portions of the rod and cone cells. Okay, so this is the external limiting membrane. Okay, now this is what surprises people about the retina when they first see its histological structure for the first time. That the rod and cones, which are the portions which actually are sensitive to light, are right at the base of the retina. They're the furthest down, basically. They're in contact with the retinal pigment epithelium. So the light is going to have to come through all of the layers on top before it will get to the rod and the cone cells. And that surprises people when they first see it. They would expect the photoreceptive portions to be right on the surface, and you wouldn't expect all of the axons to be on the surface and then the light having to get through, basically. But that's the way it is. Okay, right. So, then this next layer here, which is the nuclei of the cell bodies of the photoreceptive cells, of the rods and the cone cells, this is known as the outer nuclear layer, because you're just going to have a layer of nuclei, basically, because of course there'll be another rod cell here with its cell body here as well, and things like that. So you'll have a whole layer of nuclei, basically, and this is the outer nuclear layer, because again it's facing the outside world. It's quite an outside layer. It's a peripheral layer relatively compared to other layers of the retina. Okay, so this is an outer nuclear layer. Okay, then what you're also going to have is these photoreceptors, the rod cells and the cone cells, are now going to synapse onto bipolar neurons. Okay, so let me show this. So a bipolar neuron has two axons. One that will receive the information from the rod and the cone cells, and then another which will pass the information on. Okay, so the next cell in the pathway are these bipolar cells here. Okay, so these are bipolar neurons. And then what will happen is the bipolar neurons will pass the signal onto the next neuron along, which are the ganglion cells. Okay, so here these are ganglion cells, and then the ganglion cells will put their axons into a final layer out here, and those axons will move on the surface of the retina into the optic nerve, and then they're off to the lateral geniculate body. Okay, so this is the normal function of ganglion cells, to transfer the information in this pathway that the rod and cone cells have gathered to the brain, basically. Now, let's just talk a bit more about the different layers of the retina then. So we have this outer nuclear layer. This layer then, where we have the synapses between the bipolar neurons and the photoreceptors down here, this is called the outer plexiform layer. So this layer consists of a lot of axons basically synapsing with each other. Okay, without any cell body, so it's called the outer plexiform layer. After the outer plexiform layer, you then have another layer of cell bodies basically, which is the cell bodies of the bipolar neurons. Okay, and this is called the inner nuclear layer. Okay, so it's closer to the vitreous chamber than the outer nuclear layer, and therefore it's called the inner nuclear layer. Okay, after the inner nuclear layer, we have another plexiform layer, which just consists of axons of the bipolar neurons synapsing with axons of the ganglion cells. Okay, and this then is called the inner plexiform layer, again because we're getting closer to the innermost aspect of the eye, basically. So this is called the inner plexiform layer. And then, outside of the inner plexiform layer, you have the ganglion cell layer, okay, where the ganglion cell bodies are present. Okay, and then outside of that, we have the axons of the ganglion cells, okay, up here. And this layer is then called the nerve fiber layer, okay, because it just contains loads and loads of axons of the ganglion cells. So this is the nerve fiber layer. And all of these axons will be heading towards the place where the optic nerve begins, which is known as the optic disc. Okay, right. Then, over the top of the nerve fiber layer, what you then have is the internal limiting membrane, which demarcates the beginning of the vitreous chamber, basically. So this is the internal limiting membrane. And that now explains to us why this membrane, which was far more peripherally, was called the external limiting membrane. So this is the internal limiting membrane. Okay, so that's a bit of a reminder of the usual function of ganglion cells in the retina of the eye. Okay, so usually what happens is the light activates rod and cone cells, which then activate the bipolar neurons, which then activate the ganglion cells to fire action potentials. Okay, and that will communicate the information to the brain. However, we're going to have a special population of ganglion cells that were only discovered late in the last century, okay, which can actually respond to light themselves. So here is a new type of ganglion cell that isn't involved in this sort of a pathway, and instead is receptive to light itself. Okay, now it has a pigment that responds to light that is not found within rod or cone cells, and this is known as melanopsin. Okay, and what will happen is when light hits the melanopsin, it will activate this ganglion cell to fire action potentials, and then these action potentials will go in the axon of the ganglion cell into the optic nerve, and they're then going to make their way to the suprachiasmatic nucleus. Okay, so let me explain how these axons of these ganglion cells that are photosensitive within the retina are going to actually get to the suprachiasmatic nuclei. And by the way, these special ganglion cells are often just called photosensitive ganglion cells. So if you hear someone referring to photosensitive ganglion cells, it refers to these special ganglion cells which aren't involved in carrying signals from rod and cone cells, but instead have their own photosensitive pigment, which is melanopsin, and are going to convey the information of the light level to the suprachiasmatic nuclei. Okay, right. So let's draw a little bit of anatomy again. Here are the two optic nerves which are coming from the two eyes. Okay, so we have one eye here, we have another eye here. Okay, so here is our right eye, here is our left eye. So these axon fibers from these photosensitive ganglion cells will be coming into these optic nerves. You then have the optic chiasm, which contains the point of crossover of a huge number of axons. Okay, so usually what happens is the axons which innervate this portion of the eye on the right side, okay, so this portion in purple, which is giving you information about light sources over in this direction, okay, these will cross over onto this side to join the axons which come from this side on the left eye, basically. Okay. Meanwhile, the axons that come from this portion of the eye on the left-hand side will cross over this way to join the axons from this portion of the eye on the right-hand side, which are giving information from this side. So basically, you bring together the axons which are carrying information about a certain portion of the visual field, basically. Okay, so what's going to happen is these axons from these photosensitive ganglion cells are going to come into the optic nerve, and they're now going to get to the suprachiasmatic nuclei. Now, some of them will directly leave whilst in the optic nerve and go up to the suprachiasmatic nuclei. Some, on the other hand, will go into the optic chiasm and then go into the supraoptic nuclei. So, sorry, suprachiasmatic nuclei. The supraoptic nuclei is different to the suprachiasmatic nuclei. We're talking about the suprachiasmatic nuclei. Okay, so we have these two suprachiasmatic nuclei here, and fibers from the right eye will go into the right suprachiasmatic nucleus, and fibers from the left eye will go into the left suprachiasmatic nucleus. And by the way, the suprachiasmatic nuclei are often abbreviated to the SCNs. Now, as I say, some will go into the optic chiasm and then move into the suprachiasmatic nuclei, while some will move from the optic nerve directly into the suprachiasmatic nuclei. Now, the collective movement of all of these axons from the optic nerves and the optic chiasm into the suprachiasmatic nuclei is termed the retino-hypothalamic tract. Okay? However, do not expect to be able to go into an anatomy class and find the retino-hypothalamic tract. Okay? You will not be able to find it. It doesn't look like a little nerve that moves from the optic nerve into the hypothalamus. There are very few of these axons that are actually going from the retina into these suprachiasmatic nuclei, and you can't actually see a macroscopic structure which is moving from the optic nerve into the suprachiasmatic nuclei of the hypothalamus. Instead, it's the name for the collective movement of axons from the retina into the suprachiasmatic nuclei, but it doesn't actually physically exist in a macroscopic sense, okay? It's the name for the collective movement, but they're moving all over the place, so they're not bundled into one nice macroscopic fibre, is what I'm trying to say. Okay, and there will be two of them in principle. There's one from the right eye going into the right suprachiasmatic nucleus, and there's one from the left eye going into the left suprachiasmatic nucleus. Okay, and these will be delivering the information about the current light status of the world, basically. And this is how the suprachiasmatic nuclei get the necessary information to make sure that their internal clock is synchronized with the external clock. Okay, right, so that now concludes our discussion of the circadian rhythms.